Okay, so we're going to do things a little bit backwards today just because of the synchronous and asynchronous time. We've already gone over color vision, the different theories on how we perceive color. Now, what we're going to talk about is how light enters the eye. The good thing about this part is you have already done the asynchronous assignment of labeling the eye and what the different parts of it do. And B, you kind of just have to memorize this. You have to know the step-by-step process, but that is about it when it comes to how light enters the eye and reaches the brain. I do want you to get that diagram that I had you fill out the other day. You can print it off, that would be ideal, or you can bring it up on a separate monitor and open it up as a drawing and edit it as we go along. Okay. Or if you want to, you can just put down the step-by-step process, but you do need to know what the process is of light entering the eye and getting to the brain. Visual capture. So vision is our most dominant sense. As you saw it with the McGurk effect, which was the ba, 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 fa, 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 where even though you were hearing the same thing each time, your brain perceived it differently based on how the guy's mouth was moving. Okay, vision is our most dominant sense. It will overtake all of our other senses. This is what is called visual capture. This is how a rubber hand illusion works. So I'm going to go back and forth between the WebQuest that I had you all do the other day because I think that showing in real time is good. But so I'm going to bump back and forth between this PowerPoint and that website. So as light waves enter the eye, we have this candle right here. You're going to see the light from it. Okay, as light waves enter the eye, they enter through the protective covering on the outside, what is called the cornea. Okay, the cornea is the part of your eyeball that you can touch. If anybody's ever put in contact lenses, you're putting them on the cornea. The shape of the cornea, what it does is as this light enters the eye, it bends it to focus it to go in through the pupil. The pupil is the little black opening, little black hole in the center of our eye. The pupil is where this light enters into the eyeball. It's controlled by muscles on the outside, what is called the iris. The iris is the colored circle that surrounds the pupil. The iris's job is to dilate or expand or contract the pupil. Anybody who has had your pupils dilated before in an eye exam so that they could see better into your eye, and then you come outside and it's really, really bright out, all right, that's because your pupil is really big and it's letting in as much light as possible. Okay, so the lens's job is in dim lighting, it's going to expand your pupil. That's going to let in more light energy, making you able to see better. That's why it takes a second when you're in the dark for your eyes to adjust. Your lenses have to expand the pupil. Okay, when it's really bright outside, it dilates or it contracts it, right? It makes it smaller so that not as much light comes in so that you're not blinded by it. So once light enters the pupil, it travels through this disc that sits behind the pupil, what is called the lens. It's basically like a contact lens on the inside of your pupil. The lens's job is so that the eye can focus on whatever distance it is looking at. I'm going to go to the next slide here. It does this through a process called accommodation. When things are far away, it actually gets smaller. When things are closer, okay, it thickens, right? So it gets thinner here, thickens here for things that are close. An easy way to demonstrate this is if you turn to a wall that's relatively 10, 12 feet away from you, put your finger out in front of your face, close one of your eyes and focus on your finger. Okay, you should see your finger clearly. Now, change, you can even do it with two eyes open, change your focus to the wall behind it. Your fingers should become blurry and the wall behind it will become more clear. This is what is called accommodation. The lens changes shape, accommodation is changing. The lens changes shape based on how far away whatever it is we are looking at. Okay, so the lens focuses this light energy here through the eye, okay, onto the back of the eyeball, what is called the retina. Okay, so the retina is, we'll come to this slide in a sec, but the retina is the light sensitive back surface of our eye. All right, so I'm going to go to the WebQuest. All right, so here we go. So we'll say we have just medium brightness. All right, so we start, here's the cornea, here's the pupil right here, here's the iris right here, here's the lens. Okay, you can see how when the light comes through the lens, it focuses it. It doesn't focus it directly on the retina though. What it actually does is whatever the light energy, whatever the images you are looking at, it actually flips it upside down. So when the image hits your retina, because of the shape of the lens, you can actually do this if you go and get a spoon from your kitchen. Look at your face on the inside and the inside of the spoon, and your face will be upside down. Because of the shape of the lens, it flips that image upside down. When that light travels to the back of the eyeball, the retina, it is flipped upside down like you can see right here. The back of the retina, okay, the area of central focus, wherever it is focusing. So the retina contains different types of sensory receptors called photoreceptors. Photoreceptors are the type of receptors that transduce a neural signal to be sent to the brain. There are two types of photoreceptors on the retina. So light travels from the retina all the way to the back here. I don't know why it's on the back and not on the front. All the way to the back right here. They have these two different kinds of photoreceptors. We have what are called rods. We have many more of them. Okay. Rods are the photoreceptors that are able to pick up black, white, and gray, and they are necessary for being able to see in the dark, what's called twilight vision. Okay? They are clustered, they are mostly around the outside of our retina. Okay, so the outside areas in our retina are mostly made up of rods, which pick up white, black, gray, and are what are activated when we are in the dark. This is why we can't see colors that well when we are in the dark, because it's just these rods that are picking up the light signals. At the point of central focus on the retina, there's a spot on our retina that is called the fovea. It's right here. Okay, this is an actual picture of a retina. The fovea contains photoreceptors called cones. Cones are the photoreceptors that function in well-lit conditions and are what allow us to detect color and detail. Okay, so you can see here, all right, whatever we are focused on hits our fovea. You can remember focus, fovea. This is why whatever we are directly looking at, we can see in a lot of detail. But if you put your hands out to the sides, put your hands out like in a T, all right, and try to bring them in right on the edge of our vision, we have a harder time picking up detail and color when things are in our peripheral vision, meaning the vision that is out to the sides, right? So rods are in our peripheral vision, pick up black, white, gray. We use them in dim lighting. Cones are mostly clustered around the fovea, which is the area of central focus on the retina. And they are what allow for us to see color and detail. So, cool. All right, so here's the fovea right here. Okay, so light is projected through the vitreous humor, which is the inside of the eyeball, the liquid that's inside of the eyeball. It is projected here onto the fovea. So here's the fovea where most of our cones are. All right, here's the retina where most of our rods are. And shoot. And then this is where it gets a little bit more complicated. So these light signals trigger a chemical reaction in these rods and cones on the back of the retina. Okay, it triggers a chemical reaction in the photoreceptors. These rods and cones communicate with a type of neuron right here, what are called bipolar cells. This is where the communication between the rods and cones and the bipolar cells is where transduction occurs, is where a light signal is converted into a neural impulse. Right? So these rods and cones convert that light signal into a neural impulse, which communicates with the dendrites of the bipolar cells. These bipolar cells then communicate with the dendrites of neurons, what are called ganglionic cells or ganglion cells. These ganglion cells, their axons wind together, have bound together to form a nerve, which becomes what is called the optic nerve. You can see here in this in this diagram right here, you can see this is the neural signal that is traveling down the optic nerve right here. And I can't. Hold on, sorry. So the light signal is picked up by the photoreceptors here. It is converted into a neural impulse in the bipolar cells. And then these bipolar cells communicate with these ganglion cells, which is another type of neuron. And then the neural impulse travels down the optic nerve. From there, the optic nerve goes across what's called the optic chiasm, which is, you remember, contralateralization, which is where the two optic nerves cross to go to the other hemisphere of the brain. They travel through the thalamus, the sensory switchboard, and then from there, they travel back. The neural impulse travels back to the visual cortex of the occipital lobe to be perceived as color, to be perceived as whatever it is we are seeing at this very moment. So to put that all together, light enters through the cornea, goes through the pupil. The pupil expands and contracts by the iris. The lens is what flips this image and focuses it. On the retina, it's picked up by the rods and cones, the photoreceptors. In the bipolar cells is where transduction occurs. And then that is communicated to the ganglion cells, of which the axons form the optic nerve. From the optic nerve, it goes through the thalamus to the visual cortex, and then that is what allows for us to see. Okay, so that was quick, but that is the entire process of vision right there.